Live.
Hello, it is my pleasure to welcome each of you to the 2021 workshop on the ecology of airway infection and cystic fibrosis. My name is Nikki Lamoli, and I'm an assistant professor at the University of Iowa. Co-chairing the session is Christina Thornton from the University of Michigan, who will be working behind the scenes, handling your questions in the chat. So if at any time during the sessions you have any questions, just put them in the chat, and afterwards I will ask as many of your questions as I can during the 8-minute Q&A after each talk. So today we have an exciting lineup of speakers who will explore how interactions among bacterial, viral, and fungal communities influence the composition, function, and survival of the ecosystem during CF airway infection. They will then survey how these high-dimensional interactions are reciprocally influenced by both innate immune factors and finally CFTR modulators. So to kick things off, we have Jose Capros from Telethon Kids Institute.
Hello, everybody. My name is Jose Antonio Capros Martin, and I'm research fellow at Vaughn Respiratory Research Center. I'm working on early triggers of inflammation and infection in CF respiratory disease, with particular interest on the mechanistic pathways governing the gut-lung axis in chronic lung disease. So what I'm going to present today are some really interesting and yet unpublished results about the potential role of bile acids in the initiation and progression of CF lung disease. But before moving forward, I would like to highlight that this is the continuation of a research program that was started some time ago by Professor Fergal O'Gara and Dr. Jerry Green in Cork, Ireland, in collaboration with Professor Stephen Stick here at Telethon Kids Institute. So I have nothing to disclose. So what is the gut-lung axis? So this is a relatively new and exciting concept proposing a functional connection between these two body organs. So basically propose that molecular signals that are generated by microorganisms living in the gastrointestinal tract as a consequence of their metabolic activity, for example, short-chain fatty acids, could modulate inflammatory processes in the airways. So this is really interesting, but is there any evidence for the existence of a gut-lung axis controlling the progression of lung disease? Well, it is well known that chronic lung disorders often occur together with gastrointestinal diseases. For example, patients with different chronic respiratory disorders, for example, COPD or asthma, are more prone to develop inflammatory bowel diseases, but this could be associated with the antibiotic intake as well. And for example, asthma is a highly prevalent condition in patients with inflammatory bowel syndromes. Also, a highly prevalent intestinal comorbidity in patients with CF is gastrointestinal reflux. And interestingly, gastroesophageal reflux has been shown to be a risk factor for disease progression, as it is indicated in this forest plot. So as you can see, the use of proton pump inhibitors significantly increase the odds of developing pulmonary exacerbation. It is also significantly associated with a reduction in lung function over the time. However, it is not known if a causal relationship exists. So amongst potential components of the gastric refluxate, so molecules present in the gastric refluxate that could trigger these effects, we have shown that bile acids regulate the immune response of airway epithelial cells by repressing HIF-1 alpha signaling and by inducing the production of pro-inflammatory cytokines such as interleukin-6. And we have also demonstrated that bile acids influence both biofilm formation and antibiotic tolerance in a number of respiratory pathogens such as Pseudomonas aeruginosa or Staphylococcus aureus. Well, this sounds really interesting, but can we find bile acids in the lungs of cystic fibrosis, of patients with cystic fibrosis? And the answer is yes. These are the profiles that we found in a paper that we published last year in the BAL obtained from children with cystic fibrosis from 2 to 6 years of age. And one of the things we observed was that this concentration of bile acids were not correlated with either with the volume of saline instilled in the airways or the volume of BAL recovered, suggesting that these concentrations truly represent the bile acid load in the airways of these patients. So we can see bile acids in BAL, so what? So in this study, in the same study that we published last year, and trying to address the biological significance of bile acid detection in BAL, what we did was classifying the samples, classifying these specimens based on the bile acid profiles using an unsupervised hierarchical clustering model. And what we could see was that the samples group into 2 different clusters. So cluster 1, which is the one with higher concentration of bile acids, and cluster 2 with lower concentration of bile acids. And what we could see was that the samples in cluster 1 had higher levels of pro-inflammatory cytokines, such as interleukin beta or interleukin 6. And interestingly, despite this pro-inflammatory response, patients in cluster 1, remember the cluster with higher levels of bile acids, did not exhibit a more severe lung structural deterioration as indicated by volumetric CT scan base scores, such as, for example, including the PRAGMA-CF score. However, one of the more striking findings of this study was when we tried to address if bile acids were playing any role in the progression of lung disease. So using clinical data from the next follow-up visit, I didn't mention, but that study was a cross-sectional study. So using the follow-up, the clinical data from the next follow-up visit, we ran a linear regression model, and we could observe that patients with higher levels of bile acids, so those patients that were classified in cluster 1, were in a worse disease progression trajectory with a significant increase in the percentage of the lung with a structural disease. So it is possible that detection of bile acid could be just a consequence of a more accelerated progression of the disease. So we decided, in order to address this, we decided to evaluate associations between bile acid detection and markers of disease progression in really young patients. In this case, this was a cohort of 121 BAL specimens obtained from 1-year-old infants enrolled in the Combat CF study. So we used BAL as a surrogate of the lower airways, and we did profile the bacterial load, the bacterial and fungal microbiota using amplicon sequencing approaches, as well as the bile acids using mass spectrometry. And more importantly, we did all of these blinded to the clinical data. So one of the things, since these are really low biomass samples, one of the things we did was extracting the microbial DNA using an optimized protocol that we published last year. And as you can see here, we were able to retrieve bacterial DNA from most of the samples with a signal in the samples over the background noise generated by the negative extraction control or the instrument controls. And if we look at the profiles in this principal component analysis, we can see that the profiles in the samples were clustering away from the profiles of the controls, suggesting that they represent genuine communities. So one of the interesting things that we observed was that samples with BAL samples containing bile acids were associated with higher levels, with a higher microbial load, as you can see here in this panel, and diversity, as you can see here in this panel. And these differences were mainly driven by increases in the estimated abundance of microorganisms typically associated with the oropharyngeal niche. However, we didn't observe differences in terms of pathogen load. We also observed that samples with bile acids had distinct compositional signatures enriched in commensals and pathobionts typically associated with the upper respiratory tract, such as Streptococcus, Rotia, Haemophilus, or Gemella. So, in line with these observations, we also found that the presence of bile acids in BAL was associated with higher odds of recovering microorganisms at any density in clinical macrobiology cultures. And when we set the cutoff for positive culture to 10,000 colony forming units per mil, we observe also a significant relationship. And we also observe a more elevated load of opportunistic pathogens in samples with bile acids as well. So more interestingly, identification of bile acids was significantly associated with a higher proportion of infiltrated neutrophils and more elevated levels of interleukin-8 in BAL. Remember that these are 1-year-old infants, so we were not able to detect neutrophil elastase activity in most of the samples. However, in those samples in which we detect neutrophil elastase activity, we observe a relationship between between neutrophil elastase and bile acid detection. So we also observed that bile acid detection was associated with a significantly higher percentage of structural lung disease, but even more interesting, we observed that bile acid detection was retrospectively indicative of the severity of pulmonary exacerbations, with patients with bile acid detection having more pulmonary exacerbations during the first year of life, and also receiving more rounds of oral and inhaled antibiotics with longer treatment regimens. Interestingly, we didn't observe associations or significant association between bile acid detection and number of IV antibiotic rounds or number of days hospitalized in the case of having an exacerbation. So as a take-home message, we have observed that early in life, detection of bile acids in BAL associated with distinctive microbial communities, higher bacterial burden, higher levels of inflammatory markers, and worse clinical outcomes, suggesting that detection of bile acid in BAL represents an early biomarker of airway inflammation and microbial colonization. So finally, I would like to acknowledge all of the people that have participated in this study, especially the Combat CF study team, the participants and the families, as well as the funding agencies, the Cystic Fibrosis Foundation, the Glenn Brown Memorial Grant, the Irish Thoracic Society, as well as the Australian National Health and Medical Research Council. And of course, thanks to you for your attention. Thank you. I'm happy to take any questions you may have later.
Okay, that was a great talk. Thank you so much. So we have some questions for you, Jose, in the chat. So I'm just going to go ahead and get started. One question is, could you talk a little bit more about the clinical outcomes that you are measuring in 1-year-olds?
Well, the clinical outcomes were related to the Combat CF study, so I cannot disclose that data yet. But basically, what I was looking the outcomes we were looking at were related to the observations we found in our previous study. So basically, it was levels of inflammatory markers in BAL, so IL-8, neutrophil elastase, and markers of structural lung disease. So basically, pragma scores, number of exacerbations, and basically number of antibiotic rounds and days hospitalized, and treatments as well. So,
I have a question actually. I was wondering, in the introduction, you talked about association between asthma and other airway diseases and IBS. So do you see in those patients also increased bile acids in the airway, or is this specifically for CF patients?
Well, the bile acids, the relationship between bile acids and progression of chronic lung disease has been also shown in COPD, as far as I know. And there are some associations shown in patients with asthma in which severity of asthma seems to be related, or at least circulating levels of bile acids that seems to be associated with severity of asthma. But yes, I mean, this is what I can tell you. In regards to the relationship between chronic lung disease and disorders of the GI tract, I mean, those are associations, of course. I mean, the prevalence of inflammatory bowel syndromes in patients with chronic lung disease could be also associated with intake of antibiotics. The other case could be, well, it has been proposed that some metabolites synthesized by the gut microbiota could modulate inflammation in the lungs. But as far as I know, in humans, this hasn't been demonstrated with causal association. So.
Okay. And so do you think that the bile acids are due to microaspiration into the lungs?
Well, we didn't monitor. I mean, we cannot tell you based on I mean, this is an observational study. I cannot tell you the origin of the bile acids. We believe that this could be the case. So basically, if you look at the associations with the bacterial load and the associations with the type of bacteria that are present in those BAL samples with bile acids, you may think that probably they are coming from the upper respiratory tract through the process of macroaspiration. So yes, that could be the.
And to clarify, have you looked at the specific microbiota? Can you make those associations that there's a difference in where the bacterial communities are coming from? Or is it just bacterial load?
I look at the bacterial load, and we did the profile as well, and we did a differential analysis using a new algorithm. But basically, what we observed was that the OTUs, or the features associated with samples with bile acids in BAL, were OTUs mostly associated with the oropharyngeal cavity. So that's. Okay,
that's really interesting. And what are your thoughts on bile acids in the clinical context? Do you think this is something that we could target therapeutically, given the association in your study with structural lung damage?
Okay. The first of all, is we haven't demonstrated any causal relationship. I mean, it's really interesting that in this 1 year old, I mean, this is an association. We don't know the sense of the direction of the association. Sure. Okay. The thing is, what is really interesting is that in this study, I mean, these are 1 year old infants, so they are really, really young. And we can see that when bile acids are detected, you have inflammatory markers really high. There is some, I mean, you are expecting a mild disease in these kids, but you can see some significant differences in terms of in terms of structural lung disease and in terms of the number of exacerbations during that year. So in terms of clinical practice, I mean, what I think is that you can use these bile acids as a biomarker. So that could be, I think, the first conclusion of this paper. Obviously, we are looking at if there is any causal relationship, and that would be great, because there are ways of manipulating bile acid metabolism. I mean, there are some medications that are used to treat people with steatosis and other types of liver disease, and that can be used. I mean, you can modify bile acid metabolism through diet, and you can also modify bile acid metabolism through probiotics, for example, bacteria with bile hydroxylase activity. So, yeah.
Great, thank you. Well, thank you for a wonderful talk. And if there are any additional questions for Jose, you can connect with him through the networking modules.
Thank
So
you.
we'll go ahead and thank you. We'll move on to the second speaker in our session, who is Jack O'Connor from the Anne and Robert H. Lurie Children's Hospital of Chicago.
Hello, everyone. My name is Jack O'Connor, and I'm a research scientist in Dr. Terry Laguna's lab at Lurie Children's Hospital of Chicago. Today, I'm going to be talking to you about our work exploring the contributions of fungi to the airway microbiome in children with CF. I have no conflicts of interest to disclose regarding this work. The structure of my talk is composed of 4 parts. Initially, I will provide some background on sequencing-based approaches that have been previously used to study the lower airway microbiome and how we can potentially use those methods to characterize fungi or the microbiome in the airways of children with CF. Second, I will provide an overview of the objectives of our recent study and the methods we used. Then I'm going to present some preliminary results. Lastly, I will summarize our conclusions and describe some of our next steps. First, I will provide some background on genetic sequencing and fungal analysis. Obstructive lung disease secondary to chronic airway infection inflammation is the leading cause of morbidity and mortality in people with CF. So understanding these processes is of great clinical importance. Previous culture-based studies to better understand the CF airway have identified a few traditional CF pathogens associated with poor clinical outcomes in CF. These include Pseudomonas, Staphylococcus, Haemophilus, Burkholderia, Stenotrophomonas, and Achromobacter. However, culture studies are targeted and limit our full understanding of the CF airway microbiome. More recently, sequencing-based approaches have broadened our understanding of the lower airway microbiome. We now see the CF airway as a complex environment of dynamic polymicrobial communities. To demonstrate this, here are the culture results of the 2018 CF Foundation Registry, which show data from CF subjects across the age spectrum. You can see there were a few key players detected in culture. There was a lot of methicillin susceptible Staph aureus, colored in green. There's a lot of Pseudomonas aeruginosa in our older subjects, colored in blue. Additional traditional pathogens detected included MRSA, colored in red, and Haemophilus, colored in brown. But then when you look at 16S sequencing data, you see there are additional taxa that can be detected. This is a figure published from a paper in PLOS One by members of our group in 2016. It shows the 16S sequencing results from bronchoalveolar lavage fluid, or BALF, samples taken from 3 CF infants at 2 time points each. The y-axis is relative abundance. You can see more communities present, including Streptococcus in light blue, Veillonella in pink, and Prevotella in green, which were not traditionally associated with CF. Also, you can see community relative abundances show changes over time. In addition to bacteria, we know that fungi are present in the lower CF airway. Culture studies have identified some fungal taxa traditionally associated with CF. These include Aspergillus and Satosporium. However, the role they play in CF lung disease is not entirely understood, especially since there's so much heterogeneity in the clinical outcomes associated with fungal infections in CF. Now, in the advent of new modulator therapies, we are seeing an increase in life expectancy in adult CF patients. We're observing more complex fungal infections associated with varying clinical presentations. So it is a priority of the CF Foundation to better understand the role of fungi in CF lung disease. Traditionally, in clinic, the way that fungal infections are detected is by culture. However, fungal culture is not standardized across sites. Without standardization, varying factors like volume of the specimen processing procedures, choice of culture media, as well as incubation temperature and duration can greatly impact results. Additionally, slow growth rates and challenges in identifying fungi can present additional obstacles in diagnosing fungal infections. By developing next generation sequencing, or NGS approaches, we can provide accurate and reproducible methods to understand the lower airway fungal infections. Also, with an untargeted approach, we can broaden our understanding to include other non-traditional fungal communities present. This would facilitate further study of the relationship between fungi, lung function, and pulmonary morbidity. Now I will provide some details about the objectives of our current study and some of the methods we used. In our preliminary study, we wanted to assess the feasibility of using fungal sequencing to characterize the lower airway microbiome in CF. We wanted to also quantify the abundance of varying fungal communities in bronchoalveolar lobodulate, or BALF, obtained from subjects with CF and disease control subjects across the age spectrum. For our methods, we targeted the V6 to V8 region of the small subunit rRNA gene using polymerase chain reaction, or PCR. In addition to this 18S rRNA gene sequencing, we used quantitative PCR, or qPCR, to assess the total fungal load in our samples from CF subjects as well as disease controls. We made comparisons of total fungal load, or TFL, and the relative abundance of various fungal taxa across groups, and we also looked at age and changes with age. Now I will review some of our preliminary results. In our study, evaluation included 206 BALF samples obtained from clinically indicated bronchoscopies performed at the University of Minnesota and Children's Hospital, Colorado. Disease controls, which included any subjects who did not have a confirmed diagnosis, had a variety of underlying diagnoses and clinical indications, which is worth noting. Our CF subjects were slightly older and more female. Culture results were relatively comparable across the groups, with most samples having negative fungal cultures. CF subjects exhibited more Aspergillus. Also, one sample was positive for Candida, and the other culture data was relatively limited. When comparing qPCR results, we found that CF subjects exhibited increased total fungal load when compared to disease controls. The y-axis is the log of the copies per reaction. The CF points are colored in red and the disease control points are colored in blue. We see increased total fungal load in our CF subjects here. When assessing total fungal load across the age spectrum, we found that CF subjects also had heightened TFL across the age spectrum. On the left are box plots of the different age groups, and on the right are cubic B splines across the age spectrum. There was always increased fungal burden measured, regardless of the age of the subjects. When comparing the total fungal load to culture, we found that positive cultures exhibited slightly increased TFL. This is what we would expect. However, wider interquartile ranges of TFL in the negative cultures could be potentially attributed to a mixture of false and true culture negatives. So here we're showing that sequencing could potentially be a more sensitive method than culture. We also looked at inflammation. CF subjects had increased neutrophilic inflammation as measured by percent neutrophils, which are colored in red for the CF subjects and blue for disease controls. However, no clear relationship was observed between fungal load and inflammation. Therefore, it may be more important to understand what fungal communities are present as opposed to how much is present when considering the relationship between the microbiome and inflammation. Additionally, when compared to total bacterial load, which was also measured by qPCR, there was no clear relationship identified. Red points are the CF samples and blue points are the disease controls again. The x-axis is total bacterial load and the y-axis is total fungal load. Total fungal load does not appear to change with increasing bacterial burden. So this shows there's not really a clear relationship that we observed in this data. When looking at diversity, as measured by the Shannon Alpha Diversity Index of our fungal data, results were comparable in CF and disease control subjects across the age spectrum. So CF subjects, however, had decreased fungal diversity at older ages, which is similar to what has been seen in bacterial diversity, which usually plummets in older ages in CF. So we're seeing that generally from the fungal data, we're getting decreased diversity in our older subjects in CF. So just to summarize some of the 18S microbiome data. So 173 of the samples had sufficient fungal load for sequencing. Here's a heat map of taxa with at least 20% relative abundance in the sample set. We see diverse fungal communities in both groups. But then on the box plots on the right, when comparing Aspergillus and Candida, we did see slightly increased relative abundances in our CF group. But then also across the board in both groups, we are seeing Malassezia, which is associated with dandruff, Pleiosporalis, which is black mold, and Cladosporium, which is commonly associated with plants. So therefore, overall, we are seeing diverse fungal communities, many of which could represent potential environmental exposures. Similarly, when we split up the data by age group and look at all the relative abundances simultaneously, we are seeing very complex and diverse communities in both CF and disease controls in our different age groups. The top panel on the left is CF subjects and the bottom is disease controls. On the way left-hand side is our youngest age group, and on the right is our oldest age group. We see diverse communities in all these groups. We are also once again seeing slightly increased abundances of Aspergillus and Candida across the age groups, as is shown on the black spots here on the right. When looking at the overall abundances of the fungal taxa in the two groups, we are seeing multiple taxa identified. Once again, CF subjects had higher amounts of Aspergillus colored in red and Candida colored in brown. This is also interesting because only one sample had a positive Candida culture reported. However, as previously shown, overall, a lot of the main taxa identified in both groups were associated with dandruff, mold, and plants, including Malassezia, Pleiosporalis, and Cladosporium. Therefore, it is possible that from our sequencing, we are detecting fungal DNA inhaled into the airway from environmental sources. I will now summarize the conclusions we have made from this data and describe some of the next steps we plan to take to move our project along. In summary, while we have only gathered preliminary data, we are able to use 18S sequencing to identify complex fungal communities in BLF from CF and disease control subjects across the age spectrum. When looking at fungal load as measured by qPCR, we did find that CF subjects exhibited increased fungal burden when compared to disease controls. However, we did not observe any clear relationships between fungal load and inflammation or bacterial load. The taxa we identified included Aspergillus and Candida, which were higher in our CF subjects, and several taxa associated with potential environmental exposures. Therefore, fungal DNA detected could be from what subjects are breathing in from the environment. In terms of next steps, we do need to address some outstanding concerns and unanswered questions related to our methods. We are seeing some issues with background that complicate our analysis. Also, we're not seeing a clear relationship between fungal load and successful amplification for sequencing. With 16S bacterial sequencing, typically amplification is load dependent, and adjusting template amounts results in fairly uniform amplification. This was not the case for fungal sequencing presented here. Amplification was consistently weaker than predicted, with a small number of samples demonstrating strong amplification. And so that is another issue we need to resolve. Lastly, fungal databases have limited sequences available. That negatively impacts the coverage and sensitivity of our method. Our group has successfully resolved issues with bacterial sequencing in the past, so we feel well equipped to address these issues moving forward. We also plan to do some additional analyses, including comparing the microbiome to clinical outcomes like lung function, as well as additional inflammatory markers like pro-inflammatory cytokines and proteases in BALF. We'd also like to compare the microbiome to the microbiome as measured by 16S to explore the potential co-occurrence of lower airway bacterial communities and certain fungal communities. I want to thank several people and organizations involved in this work. We are grateful to have received funding from the American Thoracic Society, the CF Foundation, and NIH R01 grant, and the Children's Research Fund at the University of Minnesota. I want to thank our collaborators, Dr. Ryan Hunter from the University of Minnesota, as well as Dr. Kirk Harris, our microbiologist who performed the sequencing, Dr. Edith Zemanek, and Dr. Brandy Wagner, our biostatistician, all from the University of Colorado. I also want to thank Ellen Towler and Catherine Moen, who helped us get the clinical data. And then within our lab, I want to thank my PI, Dr. Terry Laguna, who has been extremely supportive, Kim Velota, our CRC, and Maddie Motlowitz, our lab tech. Lastly, I want to thank, of course, all the amazing patients and families who are willing to participate in our research. We're very grateful for your contribution. Thank you. Here are my references, and that is all. Thank you.
Thank you for a wonderful talk. We have a few questions in the chat. So I think I'll start off with a question that was about your methods. They are wondering what methods you use to extract the DNA from the fungi and the positive and negative controls that you apply when doing the extraction.
So I believe we use the QIAGEN Easy One kit to extract our DNA for this assay. when we're running the background samples, we're just including a lot of the reagents that we use for the sequencing. And so what I think's been particularly challenging is that a lot of our backgrounds were not getting a lot of successful amplification. So that's actually something we're trying to work through. And when we do some of the clustering, looking at differences between our background samples and our CF and disease controls, we're not seeing as much variability as we'd like. So I think that's sort of something we're needing to investigate a little bit further. But those are the approaches we take for extracting DNA.
Great. I was curious about the DNA that you're finding that's not typically associated with pulmonary infections, that you think it may be just DNA being inhaled. Do you think that there's a way to control for that by collecting air samples?
Yeah.
Like, do you think it's from the patients or just coming from the air?
I mean,
during the collection?
What we think, although it's still kind of preliminary in how we're trying to work through this, is that it's be coming from kind of what they're breathing in, right? It could be interesting, I think, to definitely make some comparisons, maybe with some other airway sample types to see how that may differ, to see if it could be coming potentially from the upper airway, if we want to look at like breath condensates and things like that. There could be maybe some interesting ways to approach that. But yeah, that's kind of what our thinking is on that. But there could be some interesting ways to maybe look at controlling that moving forward, especially because we're having so much issues with kind of our background samples from our reagents. That could be something interesting to look at.
And even if this is that it's not background, that there's more DNA inhaled by these patients, even if it's not from actively growing organisms, do you think that they could still be inhaling antigens that are important for inflammation?
Yeah, I mean, that's an interesting thought too, right? Because this may not necessarily be representing live cells, but could be associated with some particular antigens associated with some of these fungal taxa. But that'd be something interesting to look at too. I'm not sure kind of what would be the best way to approach that. But I think what's interesting, what we're seeing though, is the correlation between some of the loads we're seeing and the abundances we're seeing with culture, while culture is still very limited, can kind of lead us into the direction into thinking that maybe what we're picking up could be potentially reflective of active infection, just because we're seeing these correlations with culture. But at this point, still more work to do on that front.
Great, thanks. So since Aspergillus has several clinical manifestations ranging from invasive and colonization to ABPA, what are your thoughts on how to discern the various presentations through the microbiome data?
That's something, I mean, at this point, we're really trying to look at the feasibility of measuring these, right? The next step would be to kind of compare this to clinical outcomes, especially because we're seeing so much heterogeneity in some of these fungal infections, especially in older kids with CF, right? Now that we're seeing kind of in this period after these modulated therapies where we're seeing CF people living longer and longer, it's kind of been interesting to see how some of these fungal infections are arising and some of the heterogeneity we're seeing there. I think it could be, what would be really helpful, I think, for us in that case would be to obtain some of the longitudinal data. So something we're currently in the process of trying to obtain from the foundation is long-term data to see how some of these infections in these subjects, these are older samples too, how those could be, how the microbiome could be relating to some of these clinical manifestations later on. But it'll be interesting to see what comes of that. But
that's in line with another, basically a question in the chat that I think you answered about whether or not you would expect to see more dramatic differences in older or adult patients.
I mean, we see such dynamic changes in the microbiome in terms of across the lifespan in subjects living with CF. I think it's only natural we'd expect kind of similar dynamic changes you could be observing in the microbiome. Still more work we need to do in sort of gathering that data to make sense of communities that may be arising later on, or what I think will be interesting and what there's been a lot of recent work in terms of the microbiome is understanding how certain communities, how there could be crosstalk between communities that could lead subjects prone to more different chronic infections later on. It could be interesting to see how that sort of plays out with the microbiome as we gather more of this data. But yeah, I expect to see some of these changes based on these infections we're seeing just from the clinical data in older CF subjects. But it can be interesting to see as we develop more of this 18S data and get a more robust data set to see how these are changing. But
yes, we would be very interested in understanding the bacterial myco biome interactions. So
yeah,
we
I think
look
that will
forward
be interesting to see. Yeah.
Look forward to seeing those data. Thank you for a wonderful talk. We will move on to our third speaker, who is Priyati Sarwar from the University of Pennsylvania.
Good morning, / any time-appropriate greeting of your choice. I'm Priyati Sarwar, a sixth-year PhD candidate at the University of Pennsylvania from Dr. Paul Planet's lab, and I will be talking about Microbiome Db, which is a database approach to mining and integrating microbiome studies which I think will really help the CF community exploit the full potential of all the microbiome studies that are already available to it. I do not have any relationships to disclose today. The goals of my talk today are twofold. First is to demonstrate the functionalities of this Microbiome DB web platform, which is used for mining, filtering, analyzing, and visualizing 16S and shotgun metagenomic data. And I'll do that by asking my own question of it, which is to investigate the genus Prevotella in the CF, lung, and gut microbiome. So Microbiome DB is actually part of a historically free resource that's available from the University of Pennsylvania called ViewPathDB. And it is developed and maintained by the Dan Biding Lab at the Centers for Host Microbial Interactions, also at UPenn. And Dan, along with Wojtek Bazant and Anne Blevins, make up the Microbiome DB team. And this is the names and the faces you'll be interacting with if you choose to upload your data set, which is what I hope you will be encouraged to do. You can also investigate your study or your question in two ways on Microbiome DB. You can explore their publicly available data sets, which is what I will be doing, but you can also use this My Datasets feature to upload your own private data set, and then you can leverage their suite of analysis tools. So these are the questions that will be driving my talk today. And these are aimed at understanding the role of Prevotella in CF disease. And some of these questions I had while going into my exploration in Microbiome DB, and some I generated as a result of that exploration. These are the two studies that I found relevant on Microbiome DB. These are the only two CF studies. The first being, and the main focus of this talk will be the Ecocf study, which is profiling the lung microbiome of pediatric CF patients over 4 years from Dr. Paul Planet's lab. It is done using 16S RNA sequencing and is an unpublished data set. On the other hand is this baby observational and nutrition study, bonus CF from the Lucas Hoffman lab, which is a gut microbiome study, which is already published on, and it is done using shotgun metagenomic sequencing. So the reason why I study Prevotella is because it is often part of a core CF genera. It's found in high frequency. And not only are specific species of Prevotella, so the purple one being Prevotella melanogenica, found in high frequency, but multiple species of Prevotella make up this top taxa list. And the same is true when you look at a high relative abundance, that multiple species of Prevotella make up the core taxa. And when looking across a lung disease category from high to low lung disease. The role of Prevotella in disease is also quite unclear. This is just a study showing comparing oropharyngeal samples in CF and asthma patients, and both being respiratory illnesses. However, Prevotella is enriched in CF, but not in asthma. And this study shows us an association of Prevotella with increased gut health, so increased intake of dietary fiber and glucose metabolism. In vitro studies also show the potential of Prevotella in CF pathogenesis. And so this is work done from Michael Tinney's lab, which shows that the co-culture of Prevotella and Pseudomonas will have a protective effect on Pseudomonas. when exposed to antibiotics versus, when in monoculture. And then this image on this side is a representation of cytotoxicity by hemolysis, which we generated in the Planet Lab. And it shows that Prevotella strains, both within a species and between two really closely related ones, will have varied cytotoxic potential. And that is also seen in lung epithelial cell line cytotoxicity as well. So when I look at the prevalence of Prevotella in my CF studies, I see that, right off the bat, most of the samples and Ecocf respiratory samples have Prevotella in them, and only a quarter of the gut microbiome samples have Prevotella. The distribution is also vastly different, with there being a wide range of relative abundances of Prevotella in the samples, whereas in bonus CF, it's concentrated towards low relative abundance. Before I actually look at or answer these other questions that I had, I wanted to feature this one thing in Microbiome DB which made answering those questions possible, and this is their filtering feature. You can filter samples by taxon abundance or sample metadata. You can select a subset of the samples. You can build this search strategy using your parameter of choice, both from within the same study and from across studies, using this multi-step search strategy function. And then you can send those samples to the analysis tools. So when I look at the top 10 genera by of relative abundance, in Ecocf, Prevotella comes out ranked towards the top. Staphylococcus is also in the top 10 ranking in respiratory microbiomes, especially in older kids, not in infants, which makes sense since it's associated with later stage disease. And there's no Prevotella in the top 10 of the gut microbiome in bonus CF. When I stratify my respiratory samples by the presence of Prevotella and not, I find that in the samples that don't contain Prevotella, now Pseudomonas and Staphylococcus, the genus which contains the two canonically accepted pathogens of CF, are at the top of the ranking. So I was interested to see the correlation of Pseudomonas and Staph with Prevotella. So this is a quantification of what I just showed you, in that in Prevotella samples without Prevotella, there's a higher abundance of Staph, and the same is true for Pseudomonas. And then when I look at samples that contain both Prevotella and Staph, I see a slight negative correlation between the two genus, which is significant, and the same is true for Pseudomonas as well. I would like to pause here and talk about a feature of Microbiome DB that I really like, which is that even if the graph that Microbiome DB generates is not exactly what you need or in the format that you like, you can always download the data as a CSV and then export it into R, which is what I did, or Prism or Excel. So because of this negative correlation between Staph and Prevotella, I just wanted to see whether that was just a function of natural changes with age, and that appears to not be the case, as both the genus Prevotella and Staph seems to be increasing with age. So how does the alpha diversity change? So when I look at samples samples by whether or not they contain Prevotella, samples with Prevotella have higher alpha diversity, and that is true of both the respiratory microbiome and also the gut microbiome. I also wanted to see the relationship between FEV percent and alpha diversity, because that is something that's commonly studied. And I see the commonly observed trend in Ecocf as well, that at higher FEV percent, there will be higher alpha diversity. And that trend is also conserved in samples that contain Prevotella, which makes sense, as most of our samples do contain Prevotella. It's not significant, though the trend is seen in samples that don't contain Prevotella, but as you can see, there's not as many data points here. And then lastly, because I showed alpha diversity increases in Prevotella containing samples and also increases with FEV percent, I wanted to see the relationship between relative abundance of Prevotella and FEV percent, and it's showing that it remains constant across lung function, at least in Ecocf. So next, I just wanted to see how inhaled tobramycin affects Prevotella abundance, which is a question I was interested in because of the study that was recently published from the Lucas Hoffman lab, which showed that the target of maintenance inhaled tobramycin is actually predominantly non-dominant taxa, which is shown here. And in comparison, Pseudomonas, aeruginosa, and Staph aureus have very modest effects by tobramycin. So we wanted to see whether Prevotella is one of these, off-target effects of tobramycin. And not only is that, not true, Prevotella is actually enriched, though modestly so, when tobramycin is used, and as is Staph, which is I just wanted to point out. And this is a trend that we saw really only because I was exploring that FEV% and alpha diversity in Microbiome Db, and I stratified by tobramycin and saw that tobramycin use will decrease alpha diversity, which is expected. However, this trend is really only noticeable above a certain FEV percentage. So below 84% FEV value, you see that alpha diversity is resistant to change by tobramycin use. So next, I just wanted to see what was happening with Prevotella abundance in high and low FEV percent. So when I looked at the differential abundance in low FEV samples, there's no OTU with differential abundance, and Microbiome DB lets you know very politely. And high FEV percent, I see that, while there is differentially abundant taxa, Prevotella is not one of them. So some conclusions about Prevotella from this exploration. It's found in the respiratory microbiome at high frequencies with broad range of relative abundance, just not true of the gut. There is a negative correlation of Prevotella with Staph and Pseudomonas relative abundance. Samples that contain Prevotella have higher alpha diversities, and higher FEV percent values have higher alpha diversities. However, the relative abundance of Prevotella does not change across FEV percent values, and it is also modestly differentially enriched when inhaled tobramycin is used. Some conclusions about Microbiome DB is that it's a very quick and efficient exploration tool, whether or not you go in with a hypothesis. It's user-friendly, free with a low learning curve, and a very active development team. It has very easily customizable graphs, provides a standard analyses format, and also a consistent data dictionary to compare across studies. But of course, whenever you make conclusions from this sort of a cross-study analysis, the differences in study design has to be still considered. It can also be used to explore questions about underappreciated bacteria of your choice, and can be used to make novel or additional observations from published studies. And just to highlight that point, I'm just showing, something you can do is stratify very quickly by all the metadata, and you can choose scroll and just look at how relative abundance changes, which is something I did using Ecocf. You can also look at, using the correlation function tool, the correlation of individual taxa with the variable, the continuous variables that are available with your metadata. And these are my cited studies. And I would like to thank my lab, the Biting Lab, and my funding sources. I am ready for questions. Thank you so much for listening to me.
Thank you for a great talk. That's a really interesting tool that I think could have a lot of important implications for how we study microbiome in CF patients. One question is, it seems to be that there's a conflicting roles of Prevotella in CF, some showing that patients who are infected have better FEV1 and some lower. Do you have thoughts from your data on how new insights into that question from your analyses? Oh, I think you're muted.
Yeah, sorry about that. Yeah, so okay. I spent a lot of time thinking about that exact same question. I think it's hard to make conclusions like that about whether or not there are positive impacts from microbiome studies. So in the lab, I actually, another direction of my research focuses on looking at whole genome sequences. And what we find is that Prevotella species are really, really diverse. And as soon as you start adding more and more new sequences in, you'll start seeing things getting grouped with the different species. And so I think the way we think about Prevotella, because it's a little difficult to pin down in a species way, is at the genus level. So I think it will be infinitely easier to think about that question when we think about specific species. And so definitely from my studies, it seems like Prevotella melanogenica could be having pathogenic effects, but there could be Prevotella that have beneficial effects as well. And that's why those associations might conflict.
Okay, would be really interesting to be able to separate those two and have that more fine-grained detail. That's great. There's a question a little bit about the methods. So whether or not you corrected for total abundance, the comment is that the Prevotella relative abundance is lower when there are other abundant organisms. So by correcting for total abundance, do you have insight into that?
So it is normalized for total abundance, but the reason why I looked at staph and Prevotella, both, separating samples containing Prevotella not and samples that contain both Prevotella and staph, and the same with Pseudomonas, is because those things like relative abundance will, one going down will shift the other one as well, right? So there are creative ways that I decided to deal with that issue, and that was by looking at samples that contain both. And yeah, that was my way of handling how relative abundance just changes mathematically.
Okay, and there's another question on the same line of whether or not you controlled for the lack of an OTU, so no Prevotella in the alpha diversity.
Sorry, can you repeat that question?
The question is whether you controlled for the lack of an OTU in the alpha diversity. So I think in the samples that did not have Prevotella. I think this is the question.
So I think there's a base sort of in Microbiome DB, everything is like all your samples will be processed in the same way. I'm not 100% sure. 100% sure what that methodology is, though the team can certainly, provide more insights. But that is also exactly the reason why it's really hard to make any conclusions about negative Prevotella samples from Ecocf, because they're just not that many.
Great. And then when you, another question about the relative abundance. So in the samples, where you looked at the relative abundance of Prevotella, is it dominant or is that the dominant genera, or are there others that are dominant?
Yeah, it is part of the dominant genera. The most dominant would be Streptococcus. And then we see, the common other dominant genera like Veillonella rothia as well. Yeah.
And I'm of course curious about the association of Pseudomonas and whether there's an inverse relationship between, you showed the Pseudomonas and Prevotella, I think, over different age ranges, and then Pseudomonas and staph at one time point. So I'm wondering if you looked over different age ranges, do you continue to see the negative correlation between Pseudomonas and staph?
I did not do that analysis, so I can't answer that question, but I think this is my chance to plug in how useful Microbiome DB is, because if you're reading a paper and you have such questions, you can just go in and answer those yourself.
I can go answer myself. That's what you're trying to tell me. I will. We'll go take a look. We'll let you know next year. Well, thank you for a great presentation. We will move on to our next speaker. Speaker number 4 is Matthew Vaughn from Dartmouth College.
All right, let's get this set up. Hi, everyone. So my name is Matthew Bond. I am a 5th-year graduate student in Karen Adele's lab at Dartmouth College. Before I jump into my presentation, I just would like to bring up the presenter disclosure and that I have no relationships to disclose related to this presentation. So the project that I'm going to be talking to you about today focuses around bacterial biofilms and bacteriophage, and specifically how bacteriophage interact with biofilm communities and can ultimately alter the community assembly of biofilms. So for anyone unfamiliar with biofilms, just briefly, they are collections of microorganisms that are typically adhered to a surface. And they do this by secreting various proteins, carbohydrates, and extracellular DNA. They are believed to be the most predominant mode of growth for microbes in the environment, and they are extraordinarily prominent in medical infections as well. One of the challenges of biofilms in regards to medical treatment is that they're typically very robust and difficult to get rid of, especially with traditional antibiotics and even mechanical disturbances. So in order to better understand how potential future treatments, such as bacteriophage, may influence the community of microbes present in and around wounds, we have turned to a microfluidic device approach to be able to study bacterial biofilm communities. So pictured here, we have a diagram of a microfluidic device that we use on the left side of your screen. On the right, we have a picture of the device itself. So these devices are polydimethylsiloxane chips, as we call them, that are gas permeable, and they are bound to microscope cover glass. So what this allows us to do is it allows us to grow microbes and biofilms in particular in this rectangular chamber. We are able to flow fresh media in and remove the effluent from the device. And then subsequently, we can use confocal microscopy to study both the structure and composition of the communities that we grow within these devices. So while using this system, one of the first observations that was made was that E. coli biofilms, when they are sufficiently old, produce a matrix component known as curly fibers. So in this XY image here, what you can see is an E. coli biofilm. The cells are in red in this image. And you also see this white material, this white stained material that is accumulating around the cells of this biofilm. So if we take a look at this image from an orthogonal view, we can see that this white layer really covers the surface of the biofilm. So this white layer is actually the protein component in E. coli biofilms called curly fibers. And it was found that these curly fibers can actually protect the residing bacteria from a T7 bacteriophage. So it prevents the diffusion of the phage into the biofilm and subsequently prevents infection. So in this other image here, again, we have cells in red, but in magenta here, we have labeled phage. So we attached a fluorescent label to the capsid of the T7 phage, and we are able to visualize them accumulating on the surface of the biofilm, but not infecting it. I will note that curly is not stained in this image, just the phages are. So after this finding, we were curious, whether or not these phages remained active. So with biofilms being difficult to treat and phage therapies gaining prominence in the medical field these days, we wanted to know a little bit more about how these phages, if they accumulated in these biofilms, would ultimately affect the microbes around them, and ideally help to inform treatment strategies or phage treatment strategies down the road. So we kind of set out to answer three main questions with this project. The first being whether or not these phages that are trapped on the surface of the biofilm were actually active or not. Are they still able to infect incoming cells that aren't protected by the curly, or are they not able to do that? So we did this by growing a E. coli biofilm, allowing it to produce curly, and then challenging it with an invading population of E. coli. I will get into that very shortly as well, the results of that experiment. The next kind of main question that we wanted to answer was, what are some of the temporal dynamics of this system? So if we introduce new cells, are they able to integrate into the protected biofilm, or do they remain on the surface and actually remain vulnerable to incoming phages? And lastly, kind of building off of that point, if we did see some sort of protection, we wanted to know how that was occurring, basically. So like I mentioned, with our first set of experiments here, let me move myself out of the way. We grew a E. coli biofilm, the resident biofilm I will refer to it as. That is going to be your red cells here in these images, along with the inset orthogonal image. And in this instance, there was no phage pretreatment. So typically what we would do is we would grow the biofilms, and then we would either apply a vehicle control or a high titer phage prep through the chamber to coat the surface of the biofilm. So when we do perform this phage treatment, we see vastly lower biomass accumulating in this yellow invading strain. So this invading strain is just going to be an isogenic E. coli that we add in afterwards. So you can see that after the pretreatment, very few cells are able to integrate into the biofilm. And the ones that are there are actually showing a GFP reporter that we have for phage infection. So this is in stark contrast to the amount of invading biomass that we see in an untreated or control treated condition. Now, just to make sure that there wasn't any sort of physical hindrance of attachment due to the phage, we also repeated this experiment with a resistant invading population as well. So again, in this top image, we see an appreciable amount of biomass invading into the resident established biofilm. But also in the phage pretreatment condition where we did apply the phages, we still see similar amounts of invasion, implying to us that susceptibility is required for this killing to occur that we see in the wild type invasion. So if we quantify this, the results are pretty clear. When we do not have a phage treatment, we see an appreciable amount of biomass accumulating of the invading cells, again, these yellow cells. But when we do have a phage treatment, very little biomass is detectable. And those that we do see, again, are expressing this infection marker. Similarly, when we repeat the experiment with a resistant strain, we don't see a significant difference between either the control treatment or the phage pretreatment condition. So moving on from here, we wanted to see whether or not this effect of repelling biomass was due to, or if it could be influenced by time. So for this experiment, what we did was instead of doing the phage pretreatment first, instead we invaded the biofilms prior to phage treatment. So if we apply phages to the biofilms immediately following the conclusion of our invasion step, we again see very little biomass present in terms of invading cells. Now, if we wait 10 hours, we actually see an appreciable amount of biomass, again, accumulating on the resident biofilm. Excuse me. So again, if we quantify this, there's a pretty clear difference between the delays that we applied to this system. So again, if we treat the resident biofilm that has invading cells on it immediately after adding those invading cells, basically all of them are killed. And that is in contrast to if we wait 10 hours, we're seeing this protection occur for these newly arriving cells as well. And that kind of leads into the third point that we wanted to address, and that being, how is this protection occurring? So we ran a couple of experiments to help determine what might be occurring here. So briefly, I won't have time to show you all of the experiments that we did for this, but I can briefly describe some of them. So we turned to our matrix labeled strain, and we tested to see whether these invading cells themselves were actually producing curly and were able to protect themselves within that 10-hour window. But we did not see that. We did not see the invading strain producing nearly any curly throughout that time period. So we thought that maybe it's coming from the resident population instead. So what I would like to show you here is just a couple of images from a time series. So in this image, again, we have the resident biofilm in red, and we have a little pocket of invading cells in yellow. And then in cyan, we have our curly stain, so our matrix stain. So just 1 hour after the invasion step of our experiment, we see that there is matrix accumulation in the resident population, but there really isn't much surrounding those invading cells. However, if we look at the same location 10 hours after infection, what we can see is that the resident biofilm has almost like grown up. It has grown up some, and there's a lot more of this curly matrix that is now closely associated with this pocket of cells. So we can actually quantify this as well. Looking over here at this figure, this will be the mean matrix intensity for our matrix stain. And you can see that over the course of this 10 hours, the resident does accumulate more of this curly matrix around the cells. The invader also tends to increase in the amount of matrix that is associated directly around the cells. So for the invader, we see that it basically starts at 0. There's some matrix accumulation that's close to the bottom of the cells as they attach to the resident biofilm. But as we track it, we do see that matrix does increase over time, which corresponds to the images that we see here. This gradual accumulation of matrix around these invading cells. So that, it turns, is what is providing the protection for these invading cells 10 hours post-arrival, basically. So with that, I think I should be on time. And I would just like to thank my committee members, my lab members, my mentor, Carrie, all of our collaborators, all the wonderful staff and faculty members at Dartmouth who have really been instrumental in my training here. And I would also like to thank all of you for listening and the CF conference for providing me this opportunity. And of course, I would like to thank all the funding sources as well. So if you have any questions, I believe I will have a open session where you can talk to me. So feel free to reach out to me there or send me an email. Hope you guys have a great rest of the conference. Bye.
You are live.
Thanks, Matt, for a really interesting talk. We have some questions. I think the burning question is what the mechanism is. How is it that phage infection influences matrix overproduction?
So the question, if I'm understanding right, is how does the phage infection influence matrix production?
Mm-hmm.
So we haven't seen any sort of like an induction of matrix production in the presence of the phage. The biofilms do produce curly in the absence of the phage as well. So I don't really think there's really much going on in terms of inducing that matrix production. That just it seems to be you know, a byproduct that you have this dense material that actually captures the phages.
So you increased matrix production from increased growth, not that there's actually.
That's correct. Yes. Over time, yeah, they do tend to produce more. So in one of those early images that I showed, we could see the layer of curly on top of the biofilm. That does grow and spread further down into the biofilm over time if we take out the experiments a little bit further.
Okay, I see.
Mm-hmm.
And the question is, have you examined the biophysical properties of curly that is important for phage trapping? Can other EPS like PSL from Pseudomonas prevent phage invasion and subsequent invasion?
Yes, so that has actually been shown in Pseudomonas that their matrix can prevent phage diffusion, very similar to E. coli. So one of the reasons that we did go with this system was because we did have this model up and running. But the idea being that it is more broadly applicable to other microbes that are producing matrix components. Now, obviously, we don't know for sure which matrix components can block which phages. And that's certainly a burning question that we have to understand is, you know, how broadly applicable is this? Can certain matrix components only capture certain phages, or is it more of a specific kind of interaction that we're seeing?
Mm-hmm. I think that covers one of the other questions in the chat of whether matrix protection occurs for non-isogenic strains or other species.
Mm-hmm.
Sorry, I'm trying to read the other questions in the chat. How do you think the results would be different with a phage that degrades biofilms?
Degrades biofilms in terms of like secreting like proteases?
I'm not sure, or maybe just one that's more susceptible.
The phage being more susceptible, or
the biofilm?
Oh, the biofilm. So to kind of answer the first part, I imagine that there's some degree of background infection that can occur in the planktonic state. And if you are accumulating some sort of protease that could be, you know, a phage product, that certainly would influence basically that protection that you're seeing with the curly. Because typically, if there are perturbations to the curly layer, that acts basically as a hole for the phage to get in a lot of times. So we can see if there's disruption to that curly layer, propagation of the infection through that region and actually underneath the biofilm as well. So that was, what was the second part again?
I think that covers it.
Okay.
Yeah.
Sure.
Yeah. Another popular question is, what do you think the implications are of your study for phage therapy in CF patients?
Mhm. So with the advent of, I've seen a lot of studies recently about using either nebulized phages or otherwise inhaled phages that have shown very good efficacy. So what I would hope from further work, kind of down this line, is understanding how it changes the community as a whole. So from the great microbiome talks that we've heard, we know that there can be various interactions that are going on between them. And if we're treating for one, we certainly wouldn't want to be enriching for something else if there is a third member present that could provide protection, essentially. So if we know that microbe X can produce a matrix component, and that typically commingles with another organism, then maybe that would inform us a little bit more as to whether or not a treatment would be effective. But again, that's still pretty far-reaching coming from this work. We have multi-species work going on currently, so this is a very kind of foundational piece of work that we're trying to build off of to actually better answer that question.
Great, thanks. A final question about the matrix is whether the charge of the matrix influences phage diffusion.
I'm trying to recall if any charge work was done. The original paper by Lucia Vijakovic, which described this matrix blocking effect by Curley. I don't believe it looked at charge. It did look at purified. It looked at purified Curley, but I don't recall if they specifically looked at charge in that instance.
Thank you. Thanks for a really great talk. I think we'll move on to our final speaker.
Sure. Thanks for having me.
So the final speaker of this session is going to be Sarah Morgan from the University of Washington. And Sarah's going to tell us about how CFTR modulators influence all of this. So thank you very much.
Thank you for joining us. I'm Sarah Morgan, and I'm the Senior Research Scientist in the Sing Lab. I have no disclosures. Today, I want to share with you an interim analysis of the PROMISE microbiology study, which is following patients who start the new CFTR triple modulator therapy, elexacaftor, tezacaftor, and ivacaftor, here on called ETI, in order to determine how CFTR correction affects lung infections. ETI is a highly effective therapy, as many studies have shown. Here, I'm showing a box plot of sweat chloride, an indicator of CFTR function for the subjects in our study. Before ETI, almost all subjects have a high sweat chloride, but within 1 month of initiating ETI, sweat chloride drops dramatically, such that most subjects are below the diagnostic cutoff. One of the big questions with ETI is how will it affect lung infections, as lung infections are a major cause of morbidity and mortality in CF patients. To look at this, the PROMISE study collected induced sputum before and after starting ETI. This is an ongoing open-label observational study which enrolled 214 subjects with a range of disease severity as indicated by an FEV1 ranging from 29 to 130% predicted. Today, I'll be sharing with you some of the interim data from the first 6 months of this study. Despite the COVID pandemic and decreased sputum production, and thanks to the efforts of the clinical research team, we were able to get pre and at least one post-ETI sample from over 75% of enrolled subjects. Today, I'm going to show you quantitative culture results for 166 subjects who provided at least a pre and a post-starting ETI sample. Most of my talk today will focus on the changes in the microbiome using the 16S sequencing of samples from 145 subjects. But first, let's look at the quantitative culture results. Research subjects were not selected based on any prior infection criteria, so I'm going to start by showing you how cohorts who were positive for specific bacteria changed over the course of this study. Starting with Staph aureus, which is the most prevalent of the bacteria we cultured for, I'm showing you the CFU per gram sputum on the y-axis with months on ETI on the x. Each black line represents an individual subject who was positive for staph at the pre-ETI visit, with the red line indicating the average. Although a few subjects remained highly infected throughout the course of the 6 months, the majority had a decline with an average decline of after 1 month of about 2.4 logs across this group. This decline was maintained through the 6-month time point. When we looked at the cohort that started Pseudomonas positive, we see very similar results with an average decrease of 2.3 logs. And a similar pattern with Pseudomonas. I don't have time to show you, but we observed the same trend in subjects with Chromobacter and Burkholderia as well. All these decreases from pre-ETI were significant by one-way ANOVA. Intriguingly, we observed negative cultures in several subjects after starting ETI, particularly for the gram-negative organisms. The later time points collected by PROMIS may help us to determine if some subjects convert to culture negative after starting ETI. Culture is excellent for looking at a few targeted organisms, but many different organisms not conventionally defined as pathogens are found in sputum. Because of this, we wanted to use sequence-based analysis to look agnostically at all bacteria found in the sputum to determine if ETI is affecting the abundance of all organisms or just a select subset. To do this, we performed 16S sequencing on the V3 and V4 regions using standard Illumina protocols and analyzed it using the Data2 pipeline with the Silva database to identify the bacteria present. This allows us to see the composition and relative abundance of the genera present. However, because we know that all 5 of the species we've looked at decreased dramatically in abundance, we were concerned that just looking at relative abundance may not give us a full picture of how ETI is affecting the absolute abundance change. To address this, we calculated the absolute abundance by multiplying the relative abundance determined by 16S sequencing by the total 16S abundance as determined by digital drop PCR. This is a method that we have validated in prior work. Because we already know that at least some species traditionally classified as CF pathogens decrease in abundance, we decided to take a first pass by looking at the data by separating the taxa into traditional CF pathogens listed here and other bacteria commonly found in the sputum. As would be expected based on the culture results, the relative abundance, as shown as a percent on the y-axis, drops dramatically after a month on ETI and remains low throughout the remainder of the study. We see a reciprocal increase in relative abundance of the other bacteria. This could be a consequence of absolute change or simply due to it being reported as relative abundance. Because relative abundance is defined as a percentage, a decrease in one group by definition means that there must be an increase in the other group. Therefore, you could get the same pattern if traditional pathogens decreased and other genera increased, or if traditional pathogens decreased and other genera remained constant. To differentiate between these possibilities, we turn to our absolute abundance data. When we look at the calculated absolute abundance of all pathogens, we again see that the decrease in CF pathogens, which is consistent with our culture data. However, when we look at the calculated absolute abundance of the other genera, we see no discernible change in copy number after ETI. This indicates that the change in relative abundance we observed in the other group was due to a drop in pathogens. We were concerned that important changes in a few genera would be lost by this grouping. To investigate that, we looked at the calculated abundances of individual genera. Here I'm showing you the calculated abundance of the 3 most abundant genera not classified as traditional CF pathogens. We saw no significant differences in abundance pre and post ETI of Streptococcus, Prevotella, or Veillonella, or any of the other non-pathogen genera that we have looked at. However, we wanted to look agnostically at changes in all genera and to see if a large variation in abundance subject to subject was obscuring within subject changes. To do this, I'm going to focus on the 1-month time point, as this is the time point for which we observed the largest changes in abundance. Along the x-axis, I'm showing you each of the 14 genera which were present at an average abundance of greater than 1% pre-ETI. These genera are shown in order of abundance from Streptococcus, which was the most abundant, to Escherichia, which was the least. For each subject, for each genera, we calculated the log change in absolute abundance, and I'm showing you the median and the interquartile range for each genera. A negative log change indicates that the genus decreased in abundance after starting ETI. The gray shaded area represents the error inherent from the 16S measurement based upon our repeated controls. As you can see, only 4 genera decreased more than 1 log, and they were all traditional CF pathogens, as indicated by the red symbol. Both as a group and by individual genera, we see that pathogens decline after ETI, but other bacteria remain constant in response to ETI treatment. At the end, I'll come back to some potential explanations. But first, I wanted to look at changes in microbial diversity. For this, we use the Shannon Index, which takes into account both richness and evenness to investigate changes in diversity on ETI. Here, I'm showing box plots of the Shannon diversity of the bacterial community. As you can see, the median Shannon, as marked by the centroid, increased slightly but significantly after a month on ETI and remained significantly elevated throughout the 6 months of the study. However, since we saw a marked difference in the change in abundance of traditional CF pathogens, but a minimal change in other organisms, we wondered if the change in Shannon is being driven by the change in traditional CF pathogens. To address this, we computationally removed all reads belonging to traditional CF pathogens and reanalyzed the data. When we do this, we see no significant change in Shannon across any of the time points. This is consistent with the observation that abundance of traditional CF pathogens change, and the increased diversity is due to the decrease in these pathogens. In summary, quantitative culture analysis of key CF pathogens showed that most subjects experienced a decrease in CF pathogen abundance after starting ETI. The average decrease was greater than 100-fold for all 5 species studied. Further time points will allow us to determine if a subset of subjects clears infection. Analysis of the microbiome also showed the decrease in CF pathogens consistent with the culture data. When we calculated absolute abundance of other commonly identified bacteria, we found no change in these genera. We did observe a slight increase in microbial diversity, but this change appears to be driven by pathogen reduction and not changes in non-pathogens. This is preliminary data from the first 6 months of a 2-year study, but it does raise some very interesting questions, a few of which I would like to speculate on here. The first question I want to address is the infection outcome long-term? Our study on ivacaftor showed a dramatic reduction in Pseudomonas CFU over the first year, but over the subsequent years, we observed that the CFU rebounded to near original levels. I've only shown you data for the first 6 months on ETI, so it will be interesting to follow these subjects through the 2 years of this study to see if we observe a similar rebound in CFU. Furthermore, the subjects in PROMISE have a much wider range of disease severity and infection status, which may affect infection rebounds. It is also possible that since ETI is a more effective modulator, we will see more clearance and/or decreased bacterial burdens than has been seen with other modulators. Another key question that this data brings up is, if CF infections are a result of the CFTR defect, why doesn't correction of this defect allow for clearance of the infection? There are several possibilities which could help explain this. One possibility is that although we know that ETI dramatically improves CFTR function, it is possible that there are residual immune or other host defects that remain either as a consequence of damage due to infection or CFTR function not being fully restored. One key example is that we know that chronic infection in CF patients results in physical lung damage. Restoration of CFTR may not reverse the physical lung damage, and these damaged regions may harbor the infection. Another possibility is that even if modulators completely restore immune function, this is not sufficient to clear an established chronic infection. For example, host defenses are designed to protect against a small number of invading pathogens at a time, and even normal host defenses would likely be overwhelmed by large populations of pathogens. The last question I want to leave you with is, why do we observe a different response in CF pathogens versus other bacteria in response to ETI? The dramatic decrease in pathogens observed both by culture and 16S indicates that ETI is working to decrease pathogen burden, but we see no similar decrease in other genera. One possible explanation for this is that ETI restores a host function which selectively targets CF pathogens while not affecting the non-pathogens. However, studies have shown that one of the primary effects of CFTR modulators is to improve mucociliary clearance from the lungs. Because this clearance is a physical removal of sputum and all microorganisms in it, it would be expected that all organisms present in the lungs would decline upon starting ETI. Therefore, bacteria which do not decline in number may be originating from other compartments. We know that sputum must pass through the mouth in order to be collected, and that the mouth has a microbiome similar in composition to many of the genera we see here. It is therefore possible that what we are observing is that pathogens are primarily originating from lungs, and CFTR correction by ETI is increasing host clearance of the organisms in the lungs, whereas other genera are primarily originating in the mouth and upper airway and are generally unaffected by ETI. This study isn't designed to distinguish between these, and better understanding of this is going to have to rely on additional studies with invasive sampling and/or controls for upper airway sampling. And with that, I would like to thank everyone involved in this study, particularly highlight Pradeep for his mentorship and leadership, Anne and Wendy from the Sing Lab for generating most of the 16S data I showed today, Dave Nichols for all of his help making the study work and sharing his insights into the data, and most importantly, all of the participants and staff at the study sites, without whom we wouldn't have any samples. And thank you for your time.
Thank you, Sarah, for a wonderful talk. We have some questions. So do you, given the efficacy of ETI treatment and reduction of sputum production in these patients, how does that influence your ability to acquire the respiratory samples? And does it influence the quality of the sample that you get? And then what do you think, how does this promote your ability to be able to continue to follow patients over time if they're not producing sputum?
Thanks. That's actually a great question, and one that we struggle a lot with this study. The original plan of this study was to do induced sputums for everyone in order to try to address some of that in hopes that we could get more sputum from non-productive patients that way. Mm-hmm. Unfortunately, with COVID, induction of sputum became impossible at some sites. So our original samples were induced, and then some of our later samples, we have a mixture of patients who were induced, patients who were not induced, patients who were non-productive, and then patients that just weren't able to show up because of the pandemic. So that's definitely a challenge, I think, going forward for the field. We do see a lot of patients becoming non-productive. And so, you know, sputum's always been an easy way to get things, and we're just not seeing that. We're not getting as much of that from particularly some patients. Some patients are still able to produce quite a bit. And
are you able to use nasal swabs or any other samples?
Yeah, so we definitely, for the PROMISE study, we're not, but people have shown that those are really good alternatives in some cases. There can be, you know, some differences in detection between the types, but.
Sure.
Yeah.
I was wondering, is it possible to, if you look at the patients who don't respond well or who have still a significant amount of these traditional CF pathogens in the airway, can you group those patients and analyze the microbiome and identify any patterns that might inform why those patients don't respond as well?
Yeah, so we've actually looked at that a lot of ways. So we've looked at it grouping by microbiome, and what we tend to see is that those patients have a much higher percentage of pathogens, and just generally a much higher pathogen burden. We don't see a lot of differences in the non-pathogen calculated absolute abundance in those patients. But we are also looking at patient characteristics to see if sweat chloride or FEV1 or changes in sweat chloride happen. And we hope to also look at the actual genetics of the bacteria that are or are not clearing as well. So
Yeah,
that's all future work.
that would be really interesting. I look forward to hearing about that. I think maybe you just answered this question about whether there's any correlation between FEV1 change and the change in the total abundance or patterns that you see.
Yeah, we're still in the process of analyzing that, so
Okay.
I can't say quite yet.
Sure. Oh, and there's a question about whether or not you have looked at NTM, the abundance of NTM in these patients.
We have not. It's not one of the organisms that I culture for, and I don't remember seeing it on the 16S very much. So yeah, we haven't looked at that one yet.
There's a question about whether or not there are any CTs that are performed on these patients to correlate structural damage or with either lung function or the persistence of the different genera that you looked at.
Yeah, so I don't believe that there are CTs done on any of the PROMISE patients. However, Sam Durfee, a graduate student in our lab, is actually doing a research project, and she has a talk tomorrow at 11 where they've gone in and done CTs and looked at high and low damage regions, and then does BALs on those high and low damage regions to look for changes pre and post ETI. So I'd point them towards that talk.
Okay, great. Just gonna take a look at that talk. And there's a question about whether you correlated these findings to additional clinical outcomes and whether the patients who don't clear Pseudomonas tend to see less benefit with regard to exacerbations or FEV1.
Yeah, we're still looking at that. It's a lot of data to go through.
Yeah, definitely. Well, we look forward to hearing about that. Well, thank you so much for your talk. It was really interesting. And for everyone who joined us in this session, it's a really interesting conversation and a wide range of different studies on the microbiome and CF patients. So we look forward to hearing more from all of you in the future. Thanks very much.